Okay, so some important notes before we get into our lecture. One, everyone has taken exam 1 now, yay, right? So if you click on it using the Lockdown browser, we discussed this last time. Here's the information about Lockdown browser and the program if you want to download it at home. And then after you review your results, I want you to do this agreement. You don't need the Lockdown browser for that. You can just click on it. And notice in the instructions says review exam 1 results before completing this assignment. So do that, right? And then you'll complete this assignment. Oh, actually I have this one. You do have to use the Lockdown browser. I forgot. And that's my kind of double check that like you actually looked at your exam results. So this you do have to open in Lockdown browser. So remember, for Lockdown browser, you have to have everything closed on your computer. Click on it. It goes to Canvas. Log on to Canvas. Can someone get the door for me? And then go to that assignment, which for you guys, it should also just show up, right, in your things to do off to the side. So that's one of the cool features I kind of like of Canvas, right? So it'll probably be there for me, right? So see, it's right there, right? You know you need to do it. You can click right on it there, right? If you've opened and logged onto the course via the Lockdown browser, you can just click right here and go right to that assignment. So notice it's on my to-do list. I hadn't done it yet, right? So that's one thing that you guys need to get done for the 1st, which I think is Monday. So review your test, right, in Lockdown browser. Then right after you get done doing that, this is just a quick like 6-question quiz, right? It's not hard, okay? Just get it done. There's some information I want to know. So we finished up with Module 1. We're on to Module 2. I didn't realize how long it takes to really make didactic PowerPoints, right? So I had set this goal for myself to like make these interactive, more interactive PowerPoints for 2 classes. Yeah, exactly. Yeah, I really wasn't thinking. So the good news is that some of the topics from my other class carry over into this class. So I'm really concentrating on the other class right now, which you guys will get the benefit of that when we get to the next unit, which I think is probably the most difficult one for you guys. So I'm not going to post didactic, missing pieces, PowerPoints, right? I'm not going to change them to this unit. It's going to happen another semester. So you'll notice that for genetics, there's just one PowerPoint. It's the whole thing. And then I'll post the whole thing for growth and control, right? But the next module, which is our last module, I will have the really cool, like fill in the blank, get you involved PowerPoints that I had wanted to do. So the next order of business, which I think if we hit the next button, it will even take us to it. So again, animations to the links to the website pages for the book of all the animations that are available to you. Also, if you guys have the ebook, right, you can just click on it and it brings you to the animations too in the ebook. And a lot of you guys have the ebook this semester, which is cool. So in Connect 2, I wanted to bring to you guys' attention, and it's just as well that I didn't tell you before this lecture, because most of the stuff we're probably not even going to get to today. But before Tuesday's class, I highly suggest doing this bonus genetics, and it's all the animations that are important assignment. And then one of your classmates asked, and it's in that agreement assignment that I have for you guys, is that you can do these assignments as many times as you need to. And when it comes to your scores, I forget how it presents your scores to you, but one student asked, was like, I got 100, and then I didn't realize I hit submit and I got a 0. So now it's saying I have a 60% on this assignment. I go by points for one in all these assignments, so you'll notice they have points associated with them, right? So like this one is a weird points, but it's 6.25 points if you get everything right. Which you can do it over and over until you get everything right if you really want to spend the time doing that, which is great. When I download your scores from Connect, I only take your highest score, point score, for an assignment. Does that make sense to you guys? All right, so only your highest one will count. So don't worry about any of the other scores. Worry about what your highest one is for that assignment. Does that make sense to everybody? Okay. Apparently I have to say it about 12 times a semester to make sense, but it's okay. I don't have a problem repeating myself. I'm a mom. We do this constantly. You repeat yourself. Okay, so one last thing, I think. And then, of course, I'll post today's recorded lecture. I think that's it. Cool. Not too bad. So we'll go right into genetics. And so a little short review, but we've been saying this. Oh, I knew there was something else I wanted to say that I forgot. So I think I told you guys that there was a table that I saw in a book that I'm reviewing that I wanted to add for you guys. Yesterday when I was meeting with one of the book reps, I was like, ooh, I like this table in your book because it has this and this information. I was like, oh, crap. That means I have to make my own table because I want to combine information from both, right? So I can't just paste and copy or Xerox and whatever. So it's not anything you guys need to memorize per se. But I like, as you guys have experienced, I like to have real-world examples, right? So when I say that there are single-stranded RNA viruses out there, I like to be able to say, and such is that, right? So I just have a table that I'm going to put together for you guys, probably over the weekend, that gives you an example of all of them. By no means do I expect you to memorize it, but for some of us, it really helps us understand the information, right, to have real-world examples. Okay? So I'm working on that. I did not forget about it. I am working on it.
What chapter is that genetics again?
Look at your syllabus. I don't know offhand. I don't memorize chapter numbers anymore. They change. They drive me crazy. Does somebody know? Yeah, I quit putting chapter numbers in my PowerPoints because I keep changing them on me when they put out a new edition of the book. And as I said, right now we're in the process of hopefully picking a different book for you guys. So. Right, yeah, I don't even know what they would call it. Genetic variation, genetic mutation. It's 16? She says 16. There you go, 16. Mystery solved, thank you. All right, so when we were talking about viruses too, we talked about how the flow of information happens in our cells, right? The normal flow of information is DNA that's then transcribed into messenger RNA, also sometimes referred to as the transcript. So DNA and RNA are both nucleic acids, right? The same types of molecules, in the same class of molecules, right? So just as I'm talking today, you may be writing down your own transcript. You're still in the same language probably, right? English. But then that messenger RNA is read by the ribosomes. And this process is referred to as translation. Because now we're going from a nucleic acid, which is what messenger RNA is, to a different molecule. We're making proteins. What are proteins made out of? Amino acids. That's nucleic acids and amino acids, totally different molecules, right? So you do see how it's called, why it's called translation? Because we're going between different types of molecules, right? We're going from nucleic acids to amino acids, which make proteins. So this is like if you went home and took my notes and translated them into French or something. Right? So there's a reason for the terminology we use, right? We're doing a transcript, right? We're taking that message out of the nucleus for eukaryotics, right? That DNA information, we're making a transcript, we're making a message, we're taking that out, traveling to the ribosome, and that ribosome is then translating it into proteins. So there's a code involved in this, right? So people usually freak out when I put this next slide up. We know the code. We know basically the translation that's used. Okay? So this is like the secret decoder ring stuff, right? Yeah. So we know this is RNA because what abbreviation for the nucleotide or nucleic acid or nitrogenous base do you see here? You see the U, uracil, right? Uracil is only found in RNA. So we know we're looking at RNA. If we were looking at DNA, what would be there instead? The T for thymine. I almost said it even. So one other thing you'll notice here is that we have groups of 3 nitrogenous bases, or what are sometimes just referred to as bases, right? The nucleotides that make up RNA. They're read in groups of 3 by the ribosome. So that group of 3 is referred to as a codon. That codon codes for something, right? And as I said, we know the code. The code is written out in this diagram. Three of the codes tell the ribosome to stop. Apparently it needs a stop sign. We call those stop codons, of course, or nonsense codons. The rest are referred to as sense codons. They all code for an amino acid. The 3 letters you see here in blue are the amino acid 3-letter abbreviations. How many naturally occurring amino acids are present? If you add up these, which we're not going to add, you guys should know this number. 20. 20. There are 20. But notice that there are more codes, more codons, than there are amino acids. So all these codons code for the exact same amino acid. So really for this code, what's the important letters? The first 2, right? The CC. Because there are 4 different nucleotides, 4 different nitrogenous bases, but it doesn't matter which one's in the 3rd position, it's still going to be proline. There's what we call redundancy in this code. But in some cases, like over here, if it's a U or a C, it's phenylalanine. If it's an A or a G in the 3rd position, it's leucine. Totally different amino acid. And then there's one special amino acid and codon. This is methionine, and this codon is green because it tells the ribosome what? Go. Start here, right? We all associate green with go, right? That's why it's colored in green. So AUG. And for bacteria that don't do quite as much modification as we do in eukaryotic cells, that's pretty much the first amino acid of all their proteins, is methionine. And then based on the code, it will determine what comes next. So are we memorizing this code? Oh God, no. That would be insane, right? You're not geneticists, right? You're not translating this code on a daily, no. But we need to understand how this works, because how this works is why we see the changes, the quote-unquote mutations that we see. Why differences in the code will create differences in what the cell makes, what amino acid it puts there. So as I said, oops. There was a slide that I thought was here, was it? Who didn't look over his slides last night? It's okay. It's all good. All right. All that stuff that I said, I do have a slide for that. So I will put that for you guys because I think it would be good to have. All right. The reason why we look at this, why we look at genetic change in bacteria, is because it affects, a lot of times, why and how antibiotics work. Right? So it is a pretty hot topic. But in general, there are 3 different mechanisms that allow bacteria to adapt to changing environments. What happens is that they change, right? And environments are always changing, but the environment's really going to determine who survives, right? We refer to this as what? Natural selection, survival of the fittest. And this can happen because we're all different, right? Even bacteria are different like we're different. Okay? As a human population, as you look around this room, right, we are all pretty awesomely unique. Bacteria are not all that different. They have differences, right? And these differences have happened over time because of changes that happen at the DNA level, right? You look the way you do because of your DNA. In the case of me, I almost scarily look like a carbon copy of my mother. My husband says that's great because he knows what I'm going to look like when I'm, 60 years old. Apparently, I'm still going to look really freaking good. So I'm happy about that too, right? But, I have the hair that I have because of my genetics, right? I have the skin that I have because of my genetics. Bacteria have abilities because of their genetics, because of their DNA, right? So if changes happen in that DNA and they get passed on to the progeny, to the children, and that is referred to as a mutation. Now, sometimes that has kind of a negative connotation to it, right? Mutation, you think bad. Not always necessarily bad, right? So to give you a real-world example of this, there is, I think I might have mentioned this to you guys, there's a tribe in Africa that's genetically immune to HIV. Yeah. Oh, and you guys saw the thing about the bananas, right? It was on Snoop's, right? It's a fungal infection of bananas. No one's injecting bananas with HIV-infected blood. As I suspected and hoped for, it's an urban myth. Whew. Felt better about that once I looked at. Okay, so that's a genetic mutation that's good, right? That's definitely, who wouldn't want that, right? To be immune naturally because you're genetically different, right? That you can't get the form of HIV that's going around right now. It can't attach to you. How cool is that? So mutation can be really good. And so we're going to look at specific mutations, right? Like right down to the DNA level. If you substitute one base for another, what type of consequences could happen there? Right? So my husband has a genetic disease called hemochromatosis. Literally one base has been switched out in his chromosome. There's one that's off. And because of that, he has a genetic disease. Right? One nucleotide, y'all. One! That's not right. And he has a genetic disease, an inherited genetic disease from his parents. What happens if we add or remove nucleotides, right? What consequences does that have? Because remember, DNA to RNA to protein. Proteins do stuff in our cells, right? Structurally, but also very important enzymatically. All your enzymes, for the most part, are proteins. So one of his enzymes is screwed up. He accumulates blood. He accumulates more iron in his blood than is safe. Right? Has to go and literally every 4 months donate a pint of blood, and they just throw it away. They can't give it to anyone else. It's got way too much iron in it. But if that iron stayed built up in his body, it would toxify him. Right? It would poison his organs over time. and kill them. So relatively easy fix for that, right? You just got to go dump blood every 4 months, right? All kinds of fun. But it's because one enzyme isn't working, right? It's not doing its job properly. And it's literally one mutation in his DNA. One nitrogenous base wrong in the code. Hemochromatosis is what he has.
Isn't it like he calcifies or something? Like, it's all from the stigma, if he's been doing it every month for the rest of his life?
No. He hasn't, thankfully. She was saying, is it calcified? Like where they keep poking into his vein to take the blood. Hasn't yet. Thank goodness. All right. So, and we're going to look at transposable elements. So those are the main things in mutation that we're going to look at. Literally a change in the DNA, right? And what effect did those little minor changes, or what seem to be minor, have on the organism? It can lead to genetic disease, or it can lead to being immune to disease, right? Like in the case of HIV. Gene transfer. And again, remember, we're focusing on bacteria here. So these are really related to bacteria, these gene transfer events that can happen. We're going to look at transformation, where DNA can go from one bacteria to another, and we'll talk about some real-world examples with that. We're going to look at transduction, where viruses transmit genetic information from one bacteria to another. And we're going to look at conjugation, where two bacteria actually hook up with each other, sometimes referred to as bacterial sex, because they'll connect and exchange genetic information. The last one, we're not going to go into a lot of detail. I'm going to give you two real-world examples right now, and that's all we're going to talk about this, and that is the regulation of gene expression. So, for instance, in our bodies, a red blood cell makes a lot of what protein to do its job? Hemoglobin, right? Makes a ton of hemoglobin. Where is the information on how to make that hemoglobin stored? In the DNA, right? So that DNA is then transcribed into messenger RNA, and the ribosomes make hemoglobin. They make a ton of it. That's expression of genetic information, right? When you actually make what the DNA tells you how to make, you're expressing it. Now, does a liver cell need to make a ton of hemoglobin? No, that's not its job, right? It's going to make other things to do its job. A liver cell is told in the liver, right, you're a liver cell, do your liver cell job, right? But does it have in its DNA how to make hemoglobin? It does. All your cells have the exact same DNA, except what cells? For men, they're found in the testes, and for women, they're in the ovaries. Your sex cells are your gametes. What's different about them as opposed to all your somatic cells, all your other body cells? We all have 23 chromosomes, but we have how many copies of our 23 chromosomes? Two. One from mom and one from dad. And your sex cells have to be half, right? They're what we call haploid, right? So they're different from your somatic cells because they split the information in half randomly, right? They're going to take what you got from your mom and dad, and they're going to pick one copy of each. And that's it. If they do it right. If they don't, you end up with things like Down syndrome, things like that. Right? Genetic defects. Okay? So all your cells, even your liver cells, know how to make hemoglobin. But they're not told to, so they don't. Right? The environment, in that case, the liver, is dictating what those cells do. Where in the bone marrow, right, those red blood cells are like, okay, you're going to be a red blood cell, you need to make hemoglobin. You need to use that DNA. You need to express that DNA. So again, with sickle cell disease, what's the problem? Genetically, they don't have the information on how to make the right kind of hemoglobin. And you can have a minor form of sickle cell, right? You can be a carrier. You can be what we call heterozygous, because one copy you could have got from one parent is defective and the other copy was good. You have full-blown sickle cell anemia when both copies you got from both parents don't know how to make the good hemoglobin. So it coagulates in the cells and causes the cells to deform into sickle shape.
Wait, you said there was, despite being a health problem, that there was a...
There's a benefit to that, right? So what's the benefit to sickle cell? Why is it so prominent in Africa? It helps protect against a particular disease. Malaria. The protozoa that causes malaria that gets into the red blood cells can't get into the sickle cells. So if you have some sickle cells, you know, you're more resistant to infection with the plasmodium that causes malaria. So that's why that genetic defect, even though full-blown sickle cell anemia really sucks, if you're heterozygous, though, you're more resistant to getting malaria, and in Africa, of course, malaria is a really serious problem. So again, you see how environment is affecting whether something is really good or bad, or how bad it is, and why it persists, right? There's that selective pressure. And then again, environment really determines what cells are what in our bodies. Even though all the information is there, they're just going to use what they need to use. They're only going to express what they need to express. So it's under regulation, right? We regulate what genetic information we use. And it's very much environmental that will determine these things. So let's give a bacterial example, right? So we've been living in our world. Let's step back into the bacterial world. Escherichia coli, commonly abbreviated E. coli, lives in the intestines. So we're still talking about us a little bit, right? Lives in our large intestine. Loves a sugar called lactose. What food sources contain lactose? Milk, right? Milk. And things made from milk. So cheeses and yogurts and ice cream, right? All these wonderful things. There are some people that are said to be lactose intolerant. What does that mean? It makes them sick, but why does it make them sick? Anyone know? It's a sugar, right? So we would take it in, it would go through our stomach and get to our small intestine, and what do we have there in our small intestine that helps us break down our food? Enzymes, digestive enzymes, right? So is there a specific enzyme that digests lactose? Yeah, it's called lactase. Right? Some people either don't produce it or don't produce enough or stop producing that enzyme. So what happens is it doesn't get digested and absorbed in the small intestine. So then it travels on down to the large intestine, and E. coli goes, party! Because it loves lactose, y'all. It loves lactose. But it usually never sees it because you eat it first, right? You digest it, you absorb it before it even gets to the large intestine. If it makes it to the large intestine, E. coli is, I kid you not, is like, party. But does E. coli normally see lactose? No. What does it need to digest lactose? The enzymes, right? Lactase, enzymes to digest lactose.
I heard somewhere that being able, like producing the lactate or the enzyme that digests lactose is actually a mutation. We're not supposed to be able
To
digest.
yeah.
I just heard that and talking about it.
Yeah. So she was saying that evolutionary-wise, they don't believe that we were supposed to drink milk, right? But I don't believe that's true. We're mammals, right? We produce milk. We have mammary glands. Now, the fact that we drink cow's milk, now I don't know, like, does human milk not have lactose? I don't know. Does cow's milk have more lactose? I don't know. But some people believe that we mutated and then be able to produce lactase and therefore could eat lactase. So the fact that some people can't eat lactose because they don't produce lactase, they might be quote unquote normal. You see where I'm going with this one? All right. So that's a question though for us, right? I wonder if human milk has lactose and what concentration of it.
Well, I do
But-
know that. Oh, I'm sorry.
No, go
I
ahead.
know that just because
I went through a stage in my life where I was kind of vegan for a little bit, so I do know that like cow's milk is made just specifically for cows. Same thing with goats, and like we don't drink cat milk or dog milk. I mean, you see what I'm saying?
Yep.
We're only made to drink what our mom is.
Right. Yeah. Yeah. And really, a woman's milk really is dependent on what they eat. Like, so for instance, my sister was told to stop drinking milk when she was breastfeeding because her baby couldn't tolerate it, because that milk, components of the milk go into her milk that she was feeding her baby. Yeah, just like alcohol, drugs, things such as that, So, I mean, what you eat is what your baby eats, right? A version of that in your milk. So, yeah, I guess it would really depend on, right, what we eat. And that's the crazy thing, is that, we eat so many things compared to other animals, right? Like cows eat grass. At least that's all they should eat, right? And then we feed them things like oats to make them grow faster. It's not what they were intended to eat. So yeah, the real question is, I mean, we really probably were never intended to eat cow's milk, But is there other things? Where does lactose come from? Is it just in cow's milk? Might be. Right? So the fact that we can drink cow's milk is probably a mutation that happened, and so we could drink cow's milk or goat milk, all these other milks, right? Animal products, right? So back to our E. coli, lactose-loving E. coli. Lactose, the ability to digest that, who knows? Maybe that's a mutation for them too. But for the most part, they don't see lactose because we eat it first. So, do they produce the enzyme lactase all the time? No, that would be a waste, right? Why would you produce an enzyme that you will probably never use, right? Unless the person is lactose intolerant, that E. coli doesn't get to eat any lactose. But can it make it? Yeah, it has that information encoded in its DNA. So when it senses that lactose is available, like I said, he's like, woohoo, party time, right? He starts making that enzyme. He even makes more protein ports in his membrane to take in more lactose. So he starts sugar, right? Energy. He starts growing like mad crazy, right? Having babies like you wouldn't believe. So people who are lactose intolerant tend to have intestinal issues, whether it be bloating, because E. coli will sometimes ferment sugars, so therefore you get the gas production associated with it. They'll get constipation, right, where, there's so many growing they block the intestines. Or they get diarrhea, right, where it's just flowing out of the system because there's just so much in there. So much bacteria growing at such a fast rate. Make sense? Yeah. So, if you're having intestinal issues, that's one of the major ones, because again, apparently maybe we weren't supposed to eat lactose. Right? Give that a try. Cut it out of your diet. And I'll tell you, I found out I'm gluten or wheat intolerant, and come to find out part of the problem for a lot of us that have this problem is the fact that we've genetically engineered our wheat to have more gluten in it. So apparently in Europe and things like that, people don't have these types of problems because they've banned GMO, genetically modified foods. I haven't had wheat since this summer, and about a month ago, my husband and my son made cinnamon rolls, and I mean, those suckers were just screaming my name. It was horrible. I ate 2 bites, y'all. 2 bites. Had the most horrific headache I've ever had. I'm sitting on the back porch, and my husband's like, "What's wrong with you?" And I'm like, I have to tattle on myself, right? I'm like, this sucks. I'm like, I ate 2 bites of one of those cinnamon rolls in there. He's like, won't do that again, will you? I said, oh, hell no. Because the way I feel right now sucks so bad. Sucks so bad. And I've been like that most of my life. And it wasn't until this summer when I got the inflammation in my jaw taken care of my dentist. It took him 2 months too. He's like, Erica, you need to stop eating grains. You need to stop. I love bread. It was hard. It was hard. But I'll tell you, once I did it, I've never felt better in my whole life. I've never felt better. It's incredible. You really are what you eat. Eat more fresh vegetables and fruit, raw even. Raw is best, right? We screw it up when we cook it and add all this other shit to it. We do. Limit the amount of red meat you eat. I love fish though, so I'm good. I always said I'd become a chickentarian if I had to kill my own animals to eat, because I couldn't kill a cow. They're too cute, right? But I could kill me a chicken. And I could fish with the best of them, right? So, our genetics really, dictate our lives, right? Because it really depends on what we can and what we can't eat. So someone said that they were vegan and other people are vegetarian. Be very careful with those choices, y'all. Consult your doctor. Because some of us don't produce the enzymes to be vegetarians or vegans. And you actually put yourself at a disservice. You need that meat source to get some of your essential amino acids that you can't get from just fruits and vegetables alone. Right? So again, your genetics really determine. One of my colleagues here, Kendra Anspaugh, she had her DNA tested. Right? They talk about that on Ancestry all the time, right? Where you can have this company called My23, so your 23 chromosomes. You can send off a spit sample, and you can find out your heritage, but you also can find out your likelihood to have, particular genetic diseases. They test for a whole slew of stuff. It's crazy for $200. I'm considering. I was like, I want to know my son's DNA more than I want to know my own. I'm like, I think I might get him to do that and see, right? Because his dad is Cajun. I'm very curious as to that background and everything like that, and if they really are Cajun. Right? And there are certain genetic diseases that are common among ethnicity, right? So Tay-Sachs is very common in the Jewish community. Apparently breast cancer is in the French Canadian, right? The BRCA gene is very common, right? So I had my scare at the beginning of the semester. I thought I had breast cancer. That was horrible. And my grandmother was French Canadian, so there was very high chance that my biopsy could have been cancer, and it wasn't. Very happy about that. So, there's all kinds of things we can learn from our DNA nowadays. It's pretty freaking crazy. But you can also, adapt based on that, right? Obviously, if you're lactose intolerant, you're not going to eat lactose, so you don't have to go through the whole E. coli party. Or you'll take the enzymes with your food so that you digest it and they don't. Does that make sense to you guys about the expression? Right? And for us, the good news genetically is you do have 2 copies. So a lot of times, right, you're not going to run into problems unless you have, both defective copies for certain diseases. But for some of them, just one defective copy from one parent, like the BRCA gene, means that probably by now I would have already had breast cancer at my age. I'm 42. My sister had breast cancer 3 years ago at 35. So, pretty scary stuff. All right, make sense? All right, so let's all hope our genetic expression is good, right? So, we're going to identify the different types of mutations. And, the big question we're asking, how are they formed, right? How do these mutations happen? And what are their potential effects on our cells? Right? So as we already know in these cases, right, it could be a good thing. It could be like winning the lottery. You're immune to HIV. Or it could be really bad. It could be you're going to develop a particular type of cancer. Or not so bad, right? In the case of lactose intolerance, just don't eat lactose. Right? So there's this range there, right, of potential. So we're going to look specifically at base substitutions. And within base substitutions, you could have what's called a silent mutation, a missense mutation or a nonsense mutation. Notice I said this term sense before, nonsense. What was I referring to when I said nonsense? Stop codons, right? So this is going to involve stop codons. Missense is a mistake, right? A different amino acid. We're going to look at frame shifts, transposons, knockout mutations, induced mutations, chemical mutations. One of the examples for chemical mutations, there's a ton of chemicals out there, right, that cause mutations, but we're going to talk a particular one that's commonly used in scientific experimentation, known as athenium bromide. We're going to look at the effects of radiation, UV light, and X-rays, which fall under radiation. So prokaryotes are haploid, so therefore their mutations usually lead to phenotypic changes. Explain why. So first we need to make sure we know our terminology, right? So a mutation is a stable change in the DNA. It is inherited, right? So the DNA changes, it's passed on to the progeny. It doesn't get fixed. In prokaryotes, this will produce phenotypic changes. Why? Well, first of all, what is a phenotype? So genotype is the code, right, in the back? Yes. Your phenotype is the physical expression of that genetic information, right? Your genotype is the actual code, the A's, G's, T's, and C's. The phenotype is the physical expression of the genetic information, right? So in the case of hemoglobin, right, that protein is your phenotype, is the physical expression of your genetic information. What you can make from it. So sometimes it's way at the cellular level, especially when we talk about bacteria, right? They don't have curly hair or blue eyes or anything like that, right? But they have abilities, right? Do they make lactase? Do they make other proteins? All that information is in their DNA. When they express it physically, that determines their phenotype. Right? So my blue eyes, my curly hair, right? It's all encoded in my DNA. Those traits are physical expressions. They're my phenotype based on my genotype. So there was one other word in here. I'll back up. So it said prokaryotes are haploid. What does that mean?
They're half.
They're half. So, like when we talked about hemoglobin, right? You've got one copy from each of your parents on how to make hemoglobin, right? Because you have two parents, yes? Yeah. Do bacteria have moms and dads?
No.
Nope. Just mom or dad, depending on how you want to look at it.
Well,
That's
they're
it.
clone parent.
Yeah, or clone parent or something like that. But basically, they have one parent. So guess how many copies of all their genes they typically have? One. They're haploid. Just like our sex cells, they have one copy. That's it. No backup copy, one copy. Unlike us, we're what? Diploid. We have two copies. Like I said, we basically have a backup copy. So for them, if their one copy, because they're haploid, gets mutated and changed, will that affect their phenotype? What they can do? Absolutely. They don't have a backup copy. What's there is what they are, period. So we see genetic change in them faster than you would in us because they only have that one copy, right? One copy, if it's changed, their phenotype changes. Where for us, right, you don't get full-blown sickle cell anemia unless you have two bad copies. So mutations, these changes in our DNA code, right, in our genotype, They can occur spontaneously or they can be induced by chemical mutagens or radiation. Spontaneously means that basically the cell made a mistake in copying the information. Do you guys make mistakes?
Yes.
Yeah, all the time, right? Do you recognize you made a mistake?
Not all the time.
Not all the time, but sometimes, right? And you're really glad, too, sometimes, right, that you recognize you made a mistake. And then what are you going to do?
Fix it.
You're going to fix it. So do your cells have the ability, do bacterial cells have the ability to recognize mistakes and fix them?
Sometimes.
Sometimes, yeah. Sometimes. So the rate of mutation varies greatly, right? Because there's so many factors involved. How often does the cell copy? How much DNA does it have? What type of environment does it live in? I mean, so variations in the rate, the speed at which change happens can be 1 in 10,000 copies of a cell or 1 in a trillion. And one of the main reasons why the rate is so low is because all cells have cellular repair mechanisms for DNA. Because that's important stuff, right? You don't really want mistakes there. So why isn't it a perfect system? Why don't we never have a mistake? Yeah, but we have repair, but it doesn't always work, right? Some mistakes slip through. No system is a perfect system, one. Right? I don't think we can ever get to that level where a mistake is never made. Right? That would be great, but it just doesn't happen in the real world. But also, in the case of the people in Africa who are immune to, that was because of a change, y'all. They're different from the rest of us genetically because their cells, quote unquote, made a mistake. But in that case, it was what? It was a good mistake. So there is the chance you could win the lottery, right? If you go buy a ticket, there is a chance you may win. There is a chance that this change can be good change. And it's probably the reason why, evolutionarily wise, it still happens. And the fact is, like I said, there's no perfect system out there. There's no way of never making a mistake. I know we all wish and strive for that. Even at the cellular level, apparently cells wish and strive for that because mistakes can be bad. Mm-hmm.
I guess I'm trying to say, like, the immune system or something like the innate, like, they kept having exposure to it, and that's how their body started having antibodies or something like that?
Mm-mm.
Wasn't it?
No, for them, it really is a genetic change. They got lucky, and it's different, so the virus can't bind to it. Has nothing to do with exposure. Well, I should say that. Exposure really is the reason why eventually over time there would be more of them in the population, right? Because HIV is going to get everyone else if they're not careful, right? So. Make sense? Okay, so let's look at base substitution. Literally just switching out one base for another. And before I get to that, I'm going to give you guys some tips and tricks, especially for some of you guys that may want to continue on the genetic road, right, in your careers. Because this stuff is pretty cool, right? I think so. So these are just showing the nitrogenous bases in this picture. So let me fast forward a little bit to where they start labeling them. Here we go. So this is just showing the nitrogenous base part, right? You would have the sugar and the phosphate group for the whole nucleotide. But DNA, as we know, is double-stranded, right? And it has complementary base pairing. This is stuff you should know from biology class, right? A's with T's. So I always remember A, T, and T. I can't believe they're still around, but they are, right? So A binds with T. That one's pretty easy. Just remember A, T, and T. Okay. And then what binds with C? G. And C's and G's kind of look like each other, right? They're both curved, right? So it just makes sense they go together, right? But the truth of the matter is they bond with each other. In the case of A's and T's, as you can see here, it's actually 2 hydrogen bonds. And this is a relatively weak bond. It's not very strong, right? It can be pulled apart, and that has to happen, right, for DNA to be copied and for it to be transcribed into messenger RNA. G's and C's, on the other hand, have 3 bonds. But then the next thing that I want you guys to take a look at is that thymine falls into a class of nitrogenous bases. This tickle will not go away. Called pyrimidines. And they have one circular ring in their structure, where when you look over here at purines, they have 2 rings. Do you see that? So there's a little saying that actually a student taught me. Purines are A and G because angels are pure and they have 2 wings, just like purines have 2 rings. Right? So A and G, right? Adenine and guanine are both in the word angel, right? A and G. They have wings, 2 of them, just like purines, right, have 2 rings in their structure. Play on words, right, sometimes can really help. And then pyrimidines, I always think of a pyramid, you know, how it just has 1 point, so you just have 1 ring. 1 top point, I should say. Points, Scott. Need to be more specific. So this is your thymine. So if it's A and G, what do we have left? So it's cysteine and thymine. The C and the T's are the purines. And also uracil. Because uracil gets switched out for thymine in RNA. So what's the structure of double-stranded DNA?
Double helix.
Double helix, right? So like a spiral staircase. So I want you guys to imagine that in your mind right now, a spiral staircase. So imagine the steps of that staircase are these nitrogenous bases binding with each other. And the base pairing that we have is always a purine with a pyrimidine. So always 2 rings in 1 ring. So all of your steps of your spiral staircase, and this is how Watson and Crick figured it out, are 3 rings across, right? So imagine all your steps are 3 feet wide, right, for your spiral staircase. So it's this nice, even, beautiful spiral staircase. So if a carpenter came to your house and you said, oh no, well, I want a couple of the steps to be 2 feet and I want the rest of them to be 4, what would he tell you? You crazy woman, right? That would be a nightmare, right? To structurally try and get that. It's just not possible, right? To build it that way. It wouldn't be structurally sound. And this is part of the reasons why DNA works. It's structurally sound, right? This is why you have A's with T's and G's with C's. You end up with 3-foot steps for your entire spiral staircase. So again, in addition to the nitrogenous bases, which we're imagining as our steps, the banister or the railing that hooks each step together would be the sugar and phosphate that make up the nucleotide. So again, it's all uniform. It's this beautiful spiral staircase. Enzymes that read this information, that copy it into DNA, or transcribe it into messenger RNA, literally hook on to the sugar phosphate backbone and slide down it and read the nitrogenous bases as it goes. So just like a little kid will hop on a banister or a railing, right, and slide on down, so do your enzymes. So that structure, being uniform, is important to the functionality of that molecule. So what do we do to stop people from sliding down banisters? We put obstructions, right? put like little bumps on them and stuff. You see this a lot on escalators in the mall and stuff like that, right? That's basically so people don't use it as a slide. That's why those little bumps are there, y'all. As a deterrent. Do you want bumps in your DNA?
No.
No, you want sliding, right? When it comes to DNA, you want sliding. You don't want any structural deformities, right? We need it to be beautiful and functional. Make sense? So changes that really distort the DNA, like if you switch one base for another and you make that switch where you now have 4 across or 2 across, those are less likely to happen because you're going to end up with distortions in the DNA. And enzymes really pick up on that. And they're going to fix that for the most part, right? Because it's going to distort the DNA. So we have special names, right, for these different switches that can happen. I'm not going to hold you guys to that. I want you to understand this at the conceptual level, right? So let's look at this example. So what can happen here is that a change can happen in the adenine. So that instead of binding with thymine like it was a minute ago, when the enzyme comes along, it puts a cytosine there instead. Now again, we're still happy, right? Because this is still 3 rings across, right? The carpenter's not going to get upset with you. But is that the correct base pairing? No, it's supposed to be thymine, right? We have what we call a base substitution here. One base switched out for another. Here's another one, right, where changes happen to the thymine and it's binding with guanine instead of adenine. Another base substitution. So when we look at the code, right, it gets copied wrong. If the correct side doesn't get fixed, this information gets passed on to the prodigy. This is a change, so therefore it's a mutation. It's called a mutant. And as you can see, just this one change resulted in a different amino acid, right? That codon changed in such a way that it no longer codes for lysine, and wild type means the naturally occurring form of a gene for bacteria, right? That's the wild type. Ones that have significantly changed genetically, and therefore in this case also phenotypically, right? The expression is different, right? That physical expression of this genetic information is now a different amino acid. We refer to those as mutants. So, one amino acid, y'all. Is that a big deal?
At times.
It could be, right? It could be. It could be you can't do something anymore, right? Could be something you have to do to live, so now you're dead. So obviously that's not going to get passed on, right? If you mutate to that level, you're not going to pass it on. That's a lethal mutation. Those don't get passed on. The prodigy don't live. But what if it is you can't do something like digest lactose? Not the end of the world, right? There are other sugars out there. Right? That might put you at a disadvantage, right? Because if everyone else can digest lactose and it's available in the environment, they're going to outcompete you, right? They're going to win that way. But you're not necessarily going to die. Could you get a change that's really good? Yeah. Y'all, that HIV thing could be something as little as this. One base substitution. Different amino acid. Virus can't bind anymore. This happens too with bacteria and antibiotic resistance. You could change a single amino acid sometimes, say in the ribosome, and now the antibiotic no longer binds to the ribosome, and that bacteria is now resistant. Well, you can imagine in the antibiotic world, that would be pretty awesome for a bacteria, right? You're going to be the most predominant strain because you're resistant to the antibiotics. So who are we dealing with right now? The staphylococci, right? What is MRSA, or sometimes blurred together? It's actually an acronym, MRSA. Mictillison resistant Staphylococcus aureus. Micticillin is a penicillin-like drug. Staphylococcus aureus is a gram-positive bacteria. Usually penicillin-like drugs will take care of that. But this particular strain is resistant. I can't remember how or why, if it produced an enzyme that destroys it, or the protein that penicillin binds to inside the cell and interferes with its action to make peptidoglycan. I don't remember the exact mechanism. But the important thing is here, a genetic change led to this, y'all. A genetic change led to their ability to be resistant to an antibiotic. And we help perpetuate this because of the overuse of antibiotics, right? The environment is flooded with penicillin. So who's going to survive? The resistant ones. So what was our next drug of choice? Anyone know? Because guess what? We've got resistance in that too. We have VRSA now. Vancomycin. We have vancomycin-resistant Staphylococcus aureus. This will keep happening. Bacteria are going to keep changing. They're going to win the lottery every once in a while. Yeah, they're winning now. It's not pretty. And we're not helping matters in certain cases. Right? Not taking our antibiotics for the full course. When you feel better doesn't mean all the bacteria are gone, y'all. Right? Taking them when you don't need to take them. Right? When we're exposing bacteria to antibiotics when we don't need to expose them because you say have a viral infection. Right? It sucks when you go to the doctor and he says, you have a virus, I can't give you anything. Right?
I think that's mainly because they don't understand, especially if you lower that it
Yeah.
shows that it's a viral.
Yeah.
Instead of bacteria.
Yeah. So, when it comes to viruses, you're really dependent on your immune system for the most part to take care of the problem. Now, there's some antivirals out there, right? But they're targeted at viruses. Antibiotics target mainly bacteria. Right? Penicillin targets peptidoglycan. Who has peptidoglycan? Just bacteria, y'all, not viruses. Just bacteria. Make sense? Okay. So, as I said, some terminology here, right? We're not freaking out about it. But, you know, these switches, these transitions of a purine for a purine and a pyrimidine for a pyrimidine, like we saw in that animation, happen more often than transversions, where you're switching a purine for a pyrimidine. All of these, if you're switching one base for another, it's referred to as a base substitution. And we were, in fact, just looking at a single point mutation, right? A single base getting switched out. So these spontaneous mutations happen without exposure to external elements. This is just mistakes in copying the DNA. It happens. Insertions or deletions can happen. Lysing of the DNA, lesions, cuts in the DNA can happen as well. And when they try to repair it together, they don't put it together right. They'll fill in the gaps that got damaged. So again, just showing pictures like from that animation where you don't have the correct base pairing here. But again, notice you do have the 3 rings across. These are all transitions. They're not transversions. But again, not the right base pairing. And then if a base substitution is not corrected, right, this one was wrong, it can get passed on to the prodigy. We refer to this as a mutation. Specifically a base substitution. So if a single base, and that's what we've been looking at, is mutated, it's referred to as a point mutation. Just a single base has been misplaced. We can have adding or removing of nucleotides, and we can also get transposable elements that can disrupt DNA. We're going to look a little bit more deeper into this point mutation, a base substitution of just a single amino acid. And we have some terminology that relates to the outcomes that happen from just a single base being substituted at a single point. So right here, Because remember, we have some redundancy in the code. This is the normal wild type DNA. This is the normal codon in the messenger RNA read by the ribosome. Over here, there was a mistake that was made in the third position. The codon changed, but notice the amino acid did not change. It's still tyrosine. So we call this a silent mutation, because a mutation means what? Change where? Nope. Where's the change? What's this? DNA. So notice on this level right here, mutation in the DNA, in the genotype, in the code of the DNA, a change has been made. It has not been fixed. It has been passed on to the children. But phenotypically, did it change? No. But a mutation still occurred. DNA still changed. But we call this a silent mutation because the phenotype, the physical expression of that genetic information did not change. The same amino acid got put there. This can happen. The other thing that could happen is a change could happen in the DNA, a mutation in the DNA. The codon therefore changes, and the physical expression changes. We've got a different amino acid there. We call this a missense mutation. Because remember, sense is what we call the codons that code for amino acids. In this case, it was a mistake, right? Oops, we put the wrong amino acid there. It's supposed to be tyrosine, but we're coding for cytosine. What effect would this have on the organism, on the cell? Could this be good? Yeah, it's the wrong amino acid. How important is that one amino acid to that cell? Depends on the protein, right, and what it does. If that protein no longer functions because that one amino acid is wrong, that could be really bad for that cell. It can't do something it was able to do before. How important that function is to life will determine whether that cell survives or not. Or is that a disadvantage, or could it be at an advantage? Could this change be good? Yeah. Yeah. Because think about it. Think about the HIV, right? It could be something as little as a single amino acid change, and that virus can't bind anymore. But yet, the protein itself still works. So this can be good. This can be bad. This can be indifferent. It can run the whole gamut of possibilities by a single mutation and a change in amino acid in a protein. All the possibilities are there. Now this next one, instead of putting tyrosine, it put a stop codon there. The mutation in the DNA tells the ribosome to stop. We call this a nonsense mutation because the stop codons are called nonsense codons. They don't code for anything. They just tell the ribosome stop here. Could this be good? What do you think? All right, so let me give you a real-world example. Back in the Scantron test days, some of you guys might still be in, but not in my class, right? You have a 50-question test, so you got to flip the Scantron over, right? 25 on one side, 25 on the other. You don't realize, and you stop at 25 and hand in your test. Oh, you failed, right? Even if you got the first half right, that's only half. You just failed. So what are the chances that stopping could be good?
Very slim.
Very, very, very slim. Right? So with a single point mutation, we can go from really bad news with an early stop codon, right? You fail. That protein's gone. You get a mistake, which could be good, bad, the whole gamut. Or you could have a change that has no effect, silent mutation. All of these are possible with just a single change in a single nucleotide in the genotype. A single point mutation. So in the case of my husband, right, single point, y'all, in that enzyme doesn't function anymore. Cool stuff, huh?
So did you have to get testing or something?
Yeah, he's been genetically tested,
Like you
so we know specifically what his mutation is. So we're going to come to removing and adding and transposable elements. So as I said at the beginning of class, I highly suggest maybe doing that bonus assignment in Connect, right? You also have links to the animations, which will help us with some of this other stuff, right? For some of you guys, that visual added level is helpful. Try and do that before our class on Tuesday. Please make sure you fill out the agreement form, which does need the lockdown browser, after you review your test 1 results, which are up now, and I highly suggest that you do that as soon as possible, right, to learn from your mistakes.
are only things